Hey everyone, welcome to spring break. Thanks for coming to do this blast lab. It is one of our 13 labs that we have to do in the AP Biology Investigative Labs Manual, and certainly you might see it on the AP Biology test. I've asked you to do one other thing during this spring break, and that is to watch an hour-and-a-half movie on CRISPR. I think it's revolutionary and incredibly exciting and fraught with peril. And you make your own decision after you watch the movie. I will assess you to see if you watch the movie, and I do have a few things I'd like you to turn in here when you do this lab, this blast lab. I'm only looking down because I've got it in it as a piece of paper right here. Hopefully, I handed it out to you before you went away from school. Well, here it is. There's a Google Classroom pre-lab that I'd like you to look at. I don't really care necessarily if you read the Cliffs Notes book, that's fine. But I do want you to perform what I'll call the evolution assignment on the handout I've given you. And here it is. There's 4 pages to it. You'll see it. It's also on the Schoology page, and it deals with cytochrome C proteins. And basically, it's all about comparing of cladograms. And when you do that, you can try to assume some evolutionary relationships and say, oh, these things are more evolutionarily related than other things based on the analysis of one protein. Because the idea is that the DNA which creates protein mutates over time. And then when in the evolutionary tree, when species diverge from one another, there is time and opportunity for letters to change in any gene that creates any protein. And so not every protein in every every organism is going to be the same. For instance, dogs see colors differently than we do, so do snakes, so do other organisms. And the idea is that we had a common ancestor and it was able to see, but since that time, DNA has mutated and now different proteins have been created. Some had been selected against and gotten rid of, but now there are different proteins and they have different gene sequences. And we can make conclusions about evolutionary relatedness based on how close those gene sequences are. So that's what we're going to do here in this BLAST lab. BLAST is just an acronym for I have no idea what, but maybe we'll figure that out. On the Schoology page, like I said, there's 3 handouts there. One of them is an answer key for the pre-lab. You can see that, but I hope that I handed them out to you on paper. Here's what the pre-lab does look like. It just says evolution. You can see it up there in the top left corner, the molecular connection. Go ahead and try this and then take a look at the answer key, and I will be asking for this after you get back. Here's the answer key right here. Well, again, this is one of our big 13 labs, so pay attention and good luck to you. That did a lot, didn't it? Well, what is cytochrome C? You're going to see a lot of it in this lab as well as in the pre-lab. Do you remember what it is? You've heard of that term before. Where is it found? Well, here's where it's found. It's found right here in the inner membrane of mitochondria, and you know what happens there, the electron transport chain. So there it is. It's just one of these proteins that shuttle electrons that originally came from hydrogen atoms in water. Hydrogen atom that was released by NADH. I was thinking about the light reaction in photosynthesis. And it's just going to shuttle, they're just going to shuttle electrons down this chain. Now, this chain is conserved throughout all of life, higher life, not bacteria, but anything really higher than a bacteria. Protists, fungi, kingdom Animalia, and kingdom Planta also. 4 out of the 5 kingdoms have cellular respiration. And so they're going to have some form of cytochrome C shuttling electrons here in the electron transport chain. But just remember, it's not always going to be exactly the same as every other cytochrome C out there, because there's been time in these independent populations of organisms for the DNA to change and for the cytochrome C to evolve. Maybe it's better in some organisms and worse in others. And therefore, the generation of ATP from the breakdown of food is better or worse in some of these higher organisms. Maybe that's why fungus haven't really evolved to be able to ski down mountains and program computers, but maybe they can do other things that we can't. Teleport. Here's cytochrome C again. Let me show you another picture of it in all of its gory detail. There's all those alpha helices and beta pleated sheets. Here's someone's cartoon of what it might look like. Here's another person's. Maybe they aren't exactly the same. As long as they shuttle electrons down that electron transport chain, it's good. Well, just remember that what we're looking at here, when we see all those spirals and stuff, are amino acids. And you can see here's some abbreviations. Histidine, you can see tyrosine, and the number associated with those is just the number in the long primary structure of the polypeptide chain. If we were just to number all of the amino acids, it's where it would be in relation. And obviously some folding is going to take place there. Well, we know that that codons create amino acids, and you can see in this codon table or chart here, you can see abbreviations for all the amino acids. I'm going to show you a further abbreviation. This is just to further simplify things or complicate things. Amino acids each have a one-letter code. You don't need to memorize this. Just know that you're going to see these one letters and just know that they are associated with different amino acids. Well, how does the horse gene sequence for cytochrome C compare with a bunch of other organisms? That's what you're going to do today. How about comparing them to tuna, to bees, to bunny rabbits, to chicken, to frogs, the intelligent human? The idea is that we're going to look at these at cytochrome C sequences, DNA sequences for all these different organisms. And actually, what the cat's about to say here is that you can actually do any organisms that you'd like. We have these computer programs that you'll see when you watch Mr. Anderson's video at the National Institutes of Health, and they're very powerful computer programs. And the idea is that the computer is going to do the work for you. We can look up these sequences of sequences of amino acids created obviously by DNA or gene sequences in these organisms, and they're going to be a little bit different than one another. And based on those differences or similarities, we're going to try to create a phylogenetic tree here. Well, make sure that when you do this activity, you start on page 3, baseline activity. There's nothing to do. I've kind of gone over the theory on the front page here, and there's some other stuff. I'm just looking down because I have it here. I bought it long ago. It's literally a piece of paper. This is the most computer-driven laboratory we do all year. There's nothing to do except for stuff on the computer, and I don't even have a site license or anything. I just have a piece of paper I bought 5 or 6 years ago, and I wrote myself a note: protect this piece of paper. They don't sell this anymore. Don't lose it. And so anyway, okay, pull back the curtain a little bit. So here's the National Center for Biotechnology Information. Basically, you're going to use the BLAST thing over there on the right-hand side. And we're going to, there's some instructions in Mr. Anderson's video and on this piece of paper, and it will pretty much tell you what to do. Eventually, you're going to get all of these gene sequences, and then you're going to put them in here. This is a government-funded biology workbench, and basically all it does is it's a supercomputer, and it will take all of these sequences of amino acids, sorry, not gene sequences, amino acid sequences, which are governed by genes, and it will put them all together, and then it will tell you how related they are. And it will pump out something like this: an evolutionary relationship tree or phylogenetic tree. This is an example of one that one of my students got, but so here's a final product, but see anything wrong here? It just doesn't make sense. You would think that the horse and the cow would probably be more similar. And so it doesn't work. So in this final step, and I just figured this out last year, what I think kids need to do is they need to do it by hand. So apparently that website didn't work last time, and it wasn't working anyway. It's probably budget cuts. And I'm not going to say any more about that, but divide the work with your friend. And what I mean by that is go blind, comparing these sequences, and try to make your own evolutionary relatedness table. You can see one that's done here. It's probably best if I just leave. You see one that's done here, and if you're not working with a friend, don't kill yourself. Just try the best you can. Try to put the things together. It's just an exercise and an activity. And then you will turn this in to me at the end. Here's another group. They said the difference between horse and these other organisms, how many differences there were, and then they made an evolutionary tree or cladogram based upon that. Here's the 2 tips that I've learned over the years. Look for something called rec name full sequences, and at the first website page, click on the blue protein circular button, which is on the right side. Mr. Lemon's telling you that. Well, that's it for me. Good luck. I would say don't email me if you have any questions, because we can talk about it when we get back. It's just an activity. As long as you understand what's going on, I understand that there's going to be hiccups. Kids ask me every year, "What do I do here?" And of course, I'm not the most technologically savvy person, so I just kind of lie and then smile and walk away, and then they usually figure it out. Thanks, everyone.